Hello everyone. Today we will learn about restriction enzymes. This topic is in chapter Biotechnology. In this video we will learn about the restriction enzymes, their mode of action, their naming, and the type of cuts produced by these enzymes. Let's start the topic. Restriction enzymes. Restriction enzymes are also known as nucleases as they belong to the category of enzymes nucleases. Nucleases are the enzymes which specifically cut the nucleotides. As we know that DNA is the polymer of nucleotide, RNA is the polymer of nucleotide, and the enzymes which cut these nucleotides from the sugar phosphate backbone are known as nucleases. These enzymes are also known as molecular scissors because they cut the DNA at specific sites. The first restriction enzyme, particularly the endonuclease, restriction endonuclease, is Hint2. The restriction enzymes cut the DNA at specific base sequence, and these specific base sequences are also known as the recognition sequences for the enzymes. These enzymes are of two kinds. First is exonucleases and the second one is endonucleases. Exonucleases. This table is depicting the difference between exonucleases and endonucleases on the basis of their mode of action. Endonuclease separate nucleotides into two or more fragments. It cut the DNA from the center position but not from the ends. Whereas exonucleases, they remove the nucleotides one by one and they form the fragment from the ends of the DNA molecule. These will give a cut, or we can say these enzymes cut the DNA from the ends. Therefore, here it is written, they remove the nucleotides one by one and they result in formation of fragments. The second property is site of occurrence. Endonucleases result in a cleavage in DNA molecule, and this cleavage will take place in the middle of the polynucleotide chain. Whereas exonucleases will result in cleavage of the DNA molecule from the end of the polynucleotide chain. Next property is specificity. Endonucleases are sequence specific, whereas exonucleases are non-specific. Next is their function or the role. They play important role in DNA repair. They block the entry of pathogens. Whereas exonucleases, they play important role in DNA repair, stabilization, proofreading, but it will not block the entry of pathogens. This slide is about exonucleases, which remove the DNA, which cut the DNA, which remove the nucleotides from the ends of the DNA. Whereas endonucleases may cut at specific positions within the DNA. Restriction endonucleases recognize specific sequences, and these specific sequences are the palindromic nucleotide sequences. Palindrome in DNA is a sequence of base pair which reads same on two strands when the orientation of reading is kept same. Here I am going to give you an example. Suppose there is a word madam, M-A-D-A-M. If you start reading it from right to left, D will be in the center. If you read it from left to right, D will be in the center. And if you change the orientation from right to left or from left to right, you will find the first letter will be M, second will be A, and third will be D. Same kind of arrangement is found in the palindromic sequences. Here the example of palindromic sequence is given. GAATTC. If we keep the orientation of reading 5 prime to 3 prime, it is GAATTC on one strand. On the other strand of DNA, if we kept the reading frame 5 prime to 3 prime, again it will be GAATTC. So this is the example of palindromic sequence. Now, how to name restriction enzymes? Which conventions are used for naming the restriction enzymes? For naming the restriction enzymes, the first letter which we use in the name of the restriction enzyme that come from the genus in which this enzyme is found. The second letter come from the species of the prokaryotic cell from which they are isolated. Suppose here example is given E-core 1. E means Escherichia, which is representing genus, coli species, and RY13 is the strain. Here R letter is derived from the name of the strain, and the Roman number 13, or we can say here the Roman number 1. This Roman number is indicating the order in which the enzyme was isolated from that strain of the bacteria. Approx 900 restriction enzymes have been isolated from over 230 strains of the bacteria. Now, what is the mode of action of restriction enzymes? Restriction enzymes cut the DNA a little away from the center of palindromic sites, but between the same two bases on opposite strand of the DNA, and leaves a single stranded portion at the end and an overhanging stretch called sticky end on the each strand. Restriction enzymes produce two kinds of cut and result in formation of two kinds of ends. One is sticky ends, other one are the blunt ends. In next diagram, here in this diagram you can see how the restriction enzyme E-core 1 is causing a cleavage in the nucleotide sequence. Here you can see that E-core 1 is acting on a sequence GAATTC and giving a cut between G and A. How this cut is between G and A is done? In actual, it is producing a cut in the sugar and phosphodiester bond. In the backbone, there are sugar molecules which are joined to each other by the phosphodiester bond. E-core 1 give a cut between the sugar and phosphodiester bond. Thus, from this end, you will get a stretch 5' end with the G component and 3' end with CTTAA sequence. On the other strand of the DNA, we will get 5' G with, there is a cut between G and A, guanine and adenine residue, due to which you will get sticky ends. This diagram is depicting the production of sticky ends. There is an overhanging stretch, that is TAA, TTAA, and this TTAA, I am reading from 3'. If I read from 5' to 3', it will be AATT. These AATT are representing the sticky ends. Here the formation of blunt end and sticky ends formation is depicted in this diagram. Restriction enzymes, when they cleave the DNA molecule, they can produce blunt ends, they can produce sticky ends. Here the restriction enzyme Hint2 is acting on DNA molecule and producing blunt ends. These ends don't have any overhanging stretch. Here you can see the sequence is GTCGAC, and the cleavage is done by Hint2 between C and G, due to which the blunt ends are formed. Here the blunt ends are formed. No overhanging stretches there. Here you can see this is the blunt end. This is also the blunt end. But in sticky end production, here the enzyme given is BAMH1, which is acting on a sequence GGA-TCC and giving a cut between the two G residues, due to which sticky ends are produced. Here you can see after the cleavage, the two strands which you get, the two fragments which you get, these two fragments have overhanging stretch. This part is overhanging stretch. G can pair with C, but the rest part CTAG, that is the overhanging stretch. Such kinds of fragments are known as fragments with sticky ends. Again, this diagram is depicting blunt end versus sticky ends. Here the enzyme ECOR1 is digesting the sequence GAA-TTC and producing sticky ends, whereas SMA1 restriction enzyme is producing blunt ends. It is acting on the sequence CCCGGG and giving a cut between C and G, and is resulting in the production of blunt ends. Here the restriction enzyme is shown, and restriction enzyme is also known as molecular scissor. Therefore it is depicted with the help of a symbol which is representing a scissor. This restriction enzyme will cut the sugar phosphate bond which is in the backbone, and the restriction enzyme, they act on the specific recognition sites on the DNA. In this diagram, after the action of the restriction enzymes, what kind of ends are produced? Sticky ends are. That's all for today. Let's recapitulate what we have learned today. We have learnt about restriction enzymes. The enzymes belong to the category of nucleases and cut the polynucleotide chain at specific sites. Then we learnt about the types of restriction enzymes. There are two types: restriction endonucleases and restriction exonucleases. Exonucleases cut the DNA molecule from the ends, whereas restriction endonucleases cut the DNA molecule from the middle, or we can say at the specific site known as palindromic site, usually found in the center or near the center of the DNA molecule. Then we have studied about mode of action. These restriction enzymes are known as molecular scissors, and they act on, or we can say they cause a cut or cause a cleavage on the sugar and phosphate backbone of the DNA molecule, due to which the DNA fragments will be formed. Next we have done about the naming of the restriction enzymes. Restriction enzymes, during naming process, what we used to do, we will use the first letter in the naming. In the name we use the first letter, as the name of genus is used in the first letter and the species name will be used in the next letters. Then the strain is written, and then the Roman numeral is written, which depict their discovery from the strains, bacterial strains. Then type of fragments produced by restriction enzymes we have done. Two type of fragments we have done, that restriction endonucleases. In recombinant DNA technology, we will mainly use restriction endonucleases. And these endonucleases, when they cut the DNA, they produce two kinds of cuts. They produce two kinds of fragments, one with the sticky ends, and they may produce cuts in such a way that the fragment produced may have the blunt ends also. Therefore, after the action of restriction enzyme, we may get the blunt ends or we may get the sticky ends. That's all for today, students. I hope you enjoyed this. Thank you.